Good afternoon. We are students of the Instituto Tecnológico de Sonora of the Biotechnology career, and today we are going to talk about Bryostatin 1, which is a marine molecule. Bryostatin are macrocycle lactones originally isolated from the Bryostatin bubula neritina. This organism is very abundant in all oceans, and its cultivation has been attempted under controlled conditions. However, the true producer is a symbiotic bacterium that cannot survive in isolation from these organisms. Bryostatin 1 is being developed in phase 2 clinical trials by the National Cancer Institute in liaison with Bristol Myers. It seems that it's not effective without combination, which makes its synergetic effect with Taxol to be evaluated for application in lung, breast, and ovarian cancer. This compound binds with high affinity to protein C kinase, which results in anti-tumor and immunostimulating effects.
Bryostatin was discovered at Arizona State University, first insulated in the 1960s by George Petit from extracts of Bryozoan species, more exactly Bugula neritina, based on research from samples originally provided by Jack Rudolph to the group of Jonathan Hartwell's cancer drug discovery at the National Cancer Institute. Next, please. How did this happen? In 1968, Jack Rudolph published his book, The Sea Brings Forth. He appeared on the Today television show as part of the promotion of his book, brought specimens for the show, and discusses the potential for the development of natural marine drugs. The Dr. Jonathan Hartwell, who ran the U.S. National Cancer Institute, watched the show during his breakfast, and a few phone calls later, Jack Rudolph already was on the mission, collect some marine organisms and send them for analysis. 30 years passed before Bryostatin 1 was finally artificially synthesized in 1998. Next, please.
Well, the species is Bugula nerita. Bryostatin 1 is obtained from the Bryozoans Bugula nerita. They are flexibly leafy colonies with visceral branching up to 10 centimeters high. They are brown and purple in color, as you can see in the image. These organisms can be found in spores and bays attached to any type of substrate structure. It is also found in interdistal strips, coral reefs, and benthos. There are attachments to coral rocks, sponge, and sand with rocks. Its feeding has herbivorous organisms is concentrated in suspended organisms and phytoplankton. The reproduction of these species is asexual. Bryozoa are hermaphrodite and are able to generate sperm and eggs, relaxing the fruits in the celoma and thus fertilizing the eggs. Bryozoa colonies start with a single primary zooid. This primary zooid undergoes and changes for asexual germination to be able to produce a group of larval cells. Formed in the buds, and so on. The preferred scientific name is Bugula nerita. but other scientific names are Anamarchis nerita and Sertularia neritina. International common names. English is the international common name is common mungula, but the preferred common name is brown bryozoan. Well, the taxonomy. The taxonomy of Uglanarita is a Dolma eukaryota unite animalia in the age of Bryozoa, the class of Gymnolamaeta, and order Chelostomatidae, suborder of Neochilostomae, the superfamily of Bugaloidea and the family Buguloidae, the gender Bugula, and finally the species Neritine. The ecosystem. This organism is present all over the world, specifically in tropical and temperate seas. Tropical seas have an average surface water temperature of 25 centigrades. The warm waters allow to develop a great biodiversity. The temperature seas receive a minimum of 12 centigrades and have the characteristic of being very abundant in plankton, so it forecasts many migratory species. Tropical waters are located in the north and southern part of the Ecuador, with the equatorial zone being the region to warm surface temperature. How you can see in the image, the most of the place which you can find this fish is in the equatorial line. It should be noted that a symbiote called Candidatus endogubula certulia is also involved. This symbiotic carries nucleotide sequence of the PKS responsible for the biosynthesis of Bryostatin 1. You can see in the different cycles of life of the Bugula noerita, you can see the localization of Candidatus endogula certulia.
The molecular weight of the Bryostatin 1 is 905 grams mole. The name of the structure is the next.
Bryostatin synthetic diagram. In Bugula neritina, biosynthesis of bryostatin is carried out by a group of type 1 polypeptide scientists. Bry. Bry is the secondary metabolism homology of HMG CoA synthase, which is typical in bacterial primary metabolism. In the bryostatin pathway, the Bry-Air modulus catalyzes beta branching between a local acetoacetyl acceptor acyl coronary protein (HPA) and an appropriate Bray-U acetyl ASP donor. The first step involves fluorine a malonyl unit into a discrete Bray-U ASPD, waiting in the initial Bray-A module. The Bray-U product extend A is then loaded onto a Bray-Air cysteines chain for interaction with HPA. Upon interaction, Bray-Air catalyze beta branching, facilitate and aldoration between the alpha carbon of the Bray-U unit and the beta ketone, and HPA products as similar to HMGA's products in pre-metabolism. After branching, when this dehydration by a bratienoid CoA hydratase. Next.
Well, the extraction and purification. The purification of secondary or the metabolites of interest through the use of rheumatologic techniques, namely silica gel columns and Cefadex LH20 filtration gel and high efficiency liquid chromatography or LGP-LC. Structural identification of insulated metabolites using a mono and two-dimensional nuclear magnetic resonance or MRI spectroscopic techniques of proton and carbon 13, mass spectrophotometry (MS), and X-ray diffraction spectroscopy.
Bryostatin 1 acts as a potent activator of a protein called protein C kinase. In studies carried out in animal models of Alzheimer's disease, the activation of this protein results in a decrease in the accumulation of amyloid plaques in the brain. Faced with dwindling natural supplies, scientists are developing a suite of synthetic analogs as enhanced immunotherapy against cancer and HIV/AIDS eradication. Other Bryostatin-inhibit changes in protein expression can reduce symptoms of other diseases such as Parkinson and Fragile X.
Common presentation and price. They do not exist at the moment, but it is a clinical phases. Several phases. Each phase has a different purpose. And phase 1, trial is safe and look for the best way to deliver the treatment. Doctors also look for seeing that the cancer is responding to the new treatment. Phase 2. Um, Trials study whether a type of cancer responds to the phase 3. Trials whether a new treatment is better than a standard treatment. And phase 4 trials find more information on long-term benefits and side effects.
Social and ecological importance. The social importance is in the ability of this compound to improve the symptoms of different types of diseases, such as Alzheimer, a condition that increases every day worldwide and that has no cure. With Bryostatin, the symptoms can be remedied. It is also used as an anti-cancer agent. With close analogs, the effectiveness of cell therapies could be increased to a similar or better level than Bryostatin, opening the door to disease-specific optimization. That means that the effect of the disease can be less stronger. Next, please. The ecological importance. Bryosands provide habitat for many species of fluvial fish and their invertebrate prey, such as polychaete worms, amphipods, and copepods. They filter out excess food and debris particles to help keep the water clean. Next, please. And that is all for us. Thank you for listening and have a good day.
Some points are mentioned. That will be the physicochemical characteristic. The name of the structure, as you can see, is something complicated, so we are not gonna name it because we don't know the pronunciation. But is this how you can see? Is it 2 oxocyclohexacos 17 and this is a better way that we can pronounce. Which is this structure which is substituted by the hydroxy groups at the position 4, 10, and 12. An acetoxy group at position 8, methyl groups and position 9, 18, and 19. The two methoxy 2-oxoethylene groups at position 40, 14 and 12, 24. And again, sorry, but the name is some complicated for me. Octa 2 for the enoxy group at position 21. And with oxine range linking in position 6 to 10, 12 to 16, and 12 to 20 to 24. It's something important to mention that the solubility is in butanol and is in butanol and water, but principle is solubility in butanol 9.2 mg/ml. The molecular weight of Bryostatin 1 is 905 gmol. You can see this is a Bryostatin structure, the characteristic that we mentioned. And the Bryostatin biosynthetic diagram, how my parlance say, this is some complicated to understand, but we'll try to resume the biosynthetic diagram. And something important that need to understand of the explication of the phase, this phase of pharmaceutical is the reason that the Bryostatin 1 is not a product to be able to buy in a store. This is because how my partners say, this pharmaceutical, this compound or treatment is in a phase of proof. It's in the phase 2. Why, if the Bryostatin was discovered in 1962, is because this component have so many ways to treatment the different disease, how's the cancer, but no one type kind of cancer. We can this medicine can maybe can cure a different so many kind of cancer. I would say my partner Jaquelyn, other use is in the Alzheimer and SIDA, anti-cancer agent, and another disease. But the most developed or most almost finished is the Alzheimer and the Alzheimer and only that, because in the reference of cancer agent the cancer agent is in the phase 2 because there is a problem when the pharmaceutics try to obtain Bryostatin, because the problem is the biomass that you need to produce Bryostatin, because to 200 mg of Bryostatin you need to use 13 tons of biomass of Bugula nerita. And also we have the problem of the Bugula nerita that is not easy to produce in a reactor, in a fermentation. Well, a reactor, this have a need a specific condition that only can find in tropical and temperate sea. How you can see the different points that how I say is in the equatorial line. So this is all of the presentation. Thanks for the attention. This is the reference. If you have a doubt or any question, you can mail us or anything. This is all, and thanks.